You're listening to Medscape's In Discussion series on respiratory syncytial virus in adults, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Forrest Arnold. Dr. Arnold is the chief of the University of Louisville Division of Infectious Diseases and director of the Center of Excellence for Research in Infectious Diseases. He also acts as the co-editor-in-chief for the Journal of Respiratory Infections and program director for the Clinical Fellowship in Infectious Diseases. Relevant disclosures can be found with the episode show notes on medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hello, I'm Dr. Forrest Arnold, and welcome to Medscape's In Discussion series on RSV in adults. Today we'll be discussing RSV vaccines with Dr. Richard Plemper. Dr. Plemper is the Regents Professor and Distinguished University Professor at Georgia State University and Director of the Georgia State Center for Translational Antiviral Research. He specializes in pathogenesis of respiratory RNA viruses and the development of next-generation antiviral therapeutics directed against respiratory viruses of pandemic concern. Welcome to In Discussion.
Well, thank you very much. It's a great pleasure to be here, Dr. Arnold.
I think the description of your job emphasizes the topic for today, therapy for RSV. We've reviewed a few clinical case scenarios in this podcast series, but the discussion of treatment has always been short. For children, the only FDA-approved drug is palivizumab, and for adults, we have only mentioned off-label use of ribavirin. But behind the scenes of the researcher's curtain, your curtain, are a host of agents that are being investigated and are now coming close to fruition. I'd like to start with the different areas that have been and are being studied. I want to ask you about monoclonal antibodies, small molecules, and entry inhibitors, and any other you want to talk about. First, let's address monoclonal antibodies.
You mentioned palivizumab already. So palivizumab is a monoclonal antibody directed against the RSV fusion or F protein, and that's really the major protein that mediates RSV entry into new host cells. So it mediates fusion of the viral envelope membrane with a target cell membrane. Then a fusion pore opens, and through that fusion pore, the viral genetic information can enter the target cell and then start a new replication cycle. And so palivizumab was originally first developed in the late 90s. So as a monoclonal antibody, it sees or targets the RSV fusion protein, and they are a target site, an epitope that is present in two conformations of this protein, the pre- and the post-fusion conformation. When it targets in the pre-fusion conformation, then it acts neutralizing, so prevents the viral infection process, and that contributes to its therapeutic benefit. Now, the problem really with palivizumab, as you said, it is approved, but is really largely reserved for high-risk pediatric patients. So these are children either born prematurely or with underlying conditions that expose them for life-threatening RSV infection, for example, congenital heart disease. So part of the reason for that is that palivizumab is very expensive therapy and has to be administered multiple times. So it's about a month or so between individual administrations. And that really reserves it, as I said, for high-risk patients. And definitely it is a therapeutic only for high-income countries. And an RSV is really a global problem. So it was clear that we needed more therapeutic options. And very exciting, while we talk about antibodies, just very recently, the FDA advisory panel has unanimously approved or recommended approval of a new monoclonal antibody called nirsevimab that has been developed by AstraZeneca and Sanofi. And nirsevimab addresses some of these problems because it is modified in the constant region, in the FC domain, that greatly increases its half-life in the human body. So it's a therapeutic that needs to be given once to provide months-long protection of infants against RSV infection, and is highly efficacious because it sees the pre-fusion form of the F protein.
And this new drug, nirsevimab, will that be able to be used in adults?
So at the moment, the recommendation is really to protect infants because really the greatest at-risk population that are still, as I said, the infants born prematurely or with underlying conditions, at least what the FDA has considered, and it has recently been approved in Europe and in several other geographic regions in the world, is really designed to bridge this initial 6 months of age, so 6 months after birth, when infants are really most vulnerable to severe RSV infection.
Right. So it's another prevention treatment.
Correct. And of course, issue with all of the antibody treatments is that it doesn't induce or generate immune memory. So it's not a vaccine, right? Meaning that when the antibody ultimately, the titer goes down, so after a few months after administration, then the child is immunologically naive to RSV. So can be and just statistically will be infected and essentially has the primary RSV experience, which typically is more severe than later in life infections. But again, it comes after this very initial after birth high vulnerability period. So that's really the main benefit.
Okay. And what about small molecules?
Yeah, so a lot of work has been done on small molecules really over the years. 2 main areas have been targeted in the viral life cycle. One is, we already mentioned, the entry process. So these are small molecule drug-like chemical compounds that interfere, that bind to the fusion protein and prevent this conformational transition from pre-fusion to post-fusion conformation, which is essential for infection of a target cell. A number of those entry inhibitors have been explored, and several were actually advanced to clinical trials and to quite advanced stages of clinical testing. The really trailblazer was Precitabir. That's a small molecule entry inhibitor originally developed by Gilead, and that has really set kind of the roadmap for clinical testing, clinical evaluation of RSV drugs in general. So Precitabir initially performed very well, was used in an adult human challenge study. So that's if you want a study design that has very controlled conditions. It's healthy volunteers that are infected with an RSV strain, and then at defined time points, so highly controlled, are administered treatment. And Precitabir and several of the other entry inhibitors under those conditions had actually quite some promise, but were then subsequently tested in more real-world scenario conditions. Again, for example, high-risk patients in the adult group are transplant patients, so patients immune suppressed, and of course then highly susceptible to RSV and at great risk when infected by RSV to advance to severe lower respiratory tract disease. And under those more real-world scenario conditions, unfortunately, this initial batch of entry inhibitors has not provided a significant benefit to the patient. So as disappointed. However, there was a recent report at the end of last year from ArkBio out of China. That's a compound called Therapies. That's also a small molecule entry inhibitor, originally developed by Roche and then licensed by ArkBio. And they tested this compound in a phase 3 trial in infants, age 1 to 24 months. They reported significant benefit. So that really is for the entry inhibitor field, the first report of successful phase 3 clinical trial. So there is quite some excitement about this. What really the difference is between Therapies and other generation entry inhibitors is at the moment not quite clear. So the data are very new. While it's proof of concept, of course, big question is what is different in this trial compared to earlier.
Right. So let me back up and ask you a question about the Precitabir. Will that be available for adults? Is that another treatment for children?
No. So Precitabir, as I said, was even clinically tested in at-risk adult populations. So, of course, for us, if we look at the patient population that are most vulnerable, so that need therapeutic protection to prevent advanced to severe disease, and we're looking at the very young, of course, the immune compromised, I mentioned transplant patient, and older adults, because older adults, due to immune senescence, so aging of the immune system, different quality of the immune response, become more vulnerable to severe RSV. And really that is in adults age 65 and older is in the US, where we see up to 200,000 hospitalizations due to severe RSV per year and about 20,000 deaths per year. So it is a significant clinical problem. So there's a lot of excitement in RSV on the vaccine field with very exciting new developments. And we actually see also complementing the vaccine development effort, new therapeutics coming out.
Okay, so it sounds like the Procistavir, or at least the modern versions of the Procistavir, will be drugs that adults can get. That's a small molecule, while the entry inhibitors like Sarasavir that's coming out of China, or the Nirsevimab that was a monoclonal antibody, are more for children. And I think the technology that's used for these drugs for children, in a sense, is different for adults because, as you said, adults have been exposed to RSV, whereas, newborns have not. I think it's an important point to make and emphasize that you said that the small molecules will bind and prevent the pre-F protein from going to post-fusion protein. And the RSV wants it to go to post-fusion protein, right? And what's the benefit?
Correct. The fusion or F protein is what we call a metastable protein. So that's very unusual because energetically it folds when it's synthesized, not into an energy, so into a thermodynamic minimum, but it actually folds into a very high energetic state. When we discuss this with our graduate students, I use the analogy that if we imagine the free energy in a protein is like a mountain, then the F proteins are like a boulder sitting in a shallow ditch on the mountain top. That is the metastable or pre-fusion conformation. We now move or nudge this boulder a little bit, it will roll out of its ditch and then, of course, all the way down the side of the mountain till it comes to rest at the base of the mountain. That's the post-fusion conformation. And so all of this energy that this rolling boulder releases while it descends, that is the energy that is actually required for the virus to mediate the membrane fusion event. So because of that, the pre-fusion conformation, that is the one we want to target because that's the infectious virus. The post-fusion conformation, that after the refolding, so after the entry event has happened, that's essentially too late to prevent infection. So preventing this refolding is key. But for many years, a real challenge was that if we want to study the fusion proteins and express them, that spontaneously, when expressed in artificial systems outside of the context of the virus, or if they're extracted from the virus, then they immediately assume this energy low post-fusion conformation. And so that created a major obstacle that essentially the target we're most interested in, pre-fusion F, structurally for many decades of RSV research and research on related viral pathogens was undefined. And that really in the last decade or so has drastically changed through structural information of the pre-fusion protein, which has really driven not only vaccine development, but also therapeutics development enormously.
I like this idea of a dead volcano with a crater at the top and the boulder sitting up there, and that's representing the pre-fusion state. And if you nudge it, it'll roll down that mountain, and there's a lot of energy released, and then it finally comes to rest as the post-fusion state. I can imagine a physician in a clinic wondering what's the relevance of that, but there's two things that would come to light, and that is everybody's known that RSV is terrible in children, but now there's been an acknowledgement that it's also terrible for very old adults. And the work that's being done to find therapeutics for children is rubbing off, and adults are being the beneficiary of that in some regard as well. Another one I'd like to mention is molnupiravir, and we're familiar with that to some degree because it's FDA approved for COVID-19 when ritonavir-boosted nirmatrelvir and remdesivir are not available. But what is the RSV story about this drug?
Yeah, so molnupiravir, my lab has worked on molnupiravir and related drugs for really several years now, way before it was actually called molnupiravir. So molnupiravir is a prodrug, meaning it's the orally bioavailable form of N4-hydroxycytidine. So that's a nucleoside analog, meaning an analog of the building blocks of genetic information. And when incorporated into the viral genome, then introduces spontaneous viral mutagenesis that very rapidly results in collapse of the viral population. So it destroys the viral genome. Now, when we started working with molnupiravir, or rather its precursor, so its active metabolite, then originally what we actually targeted were influenza virus and respiratory syncytial virus. And so one of the first publications we had on the topic of this metabolite of molnupiravir demonstrates that it's actually very efficacious in animal models at shutting down RSV replication. And then subsequently, when we had together with our colleagues at Emory developed what is now called molnupiravir, we actually targeted initially as what we thought pre-pandemic was the greatest challenge, and that was the influenza virus. That happened in 2019 when an influenza trial was actually set up. And then, of course, we all know the zoonotic transmission event in Wuhan and the global pandemic, and that has redirected many things, including molnupiravir against SARS-CoV-2. But right now, Merck is actually running in adults a clinical trial, a challenge trial with molnupiravir for the treatment of RSV. So as you said, we talked about the antibodies and, for example, nirsevimab, which are really targeted at the infants. Now, molnupiravir for treatment of RSV is kind of the opposite. This would be not for the pediatric patient population, but molnupiravir would be used to protect the elderly against life-threatening RSV infection. Not as a prophylactic or not like an antibody that we give prophylactic. But we shouldn't forget, right? So in the elderly, RSV typically starts as upper respiratory infection with the classical symptoms of upper respiratory disease. Advanced to severe disease entails that the virus progresses to the small airway. So we have bronchiolitis, viral pneumonia. Virus is then very mucogenic. So a lot of mucus induction, bronchial obstructions. That is the life-threatening state. The idea is to prevent the advance from upper respiratory disease to viral pneumonia, so to lower respiratory tract infection. And that really will require that not only we have efficacious treatment, but also that time is of the essence. So what we call the therapeutic time window for efficacious treatment is definitely short for these acute RNA viral diseases like RSV disease, meaning the earlier, really with the positive diagnosis of RSV, the earlier treatment is started, the more therapeutic benefit can be anticipated.
Right. So a stubborn older adult that thinks they just have another cold, maybe doing themselves harm by not presenting sooner. I think in the hospital, we've done a good job by putting RSV on a rapid viral panel that just has influenza, COVID, and RSV on it. But as an outpatient, that's an area where we could improve because there's rapid COVID tests, even the home ones, and influenza rapid tests in the doctor's office, but not so much RSV rapid test. So if this could be performed, then those adults could be cared for sooner and maybe even given one of these therapeutic agents if it's indicated to outpatients as well. Correct. Yeah, I mentioned COVID-19. What did that do to the RSV research environment and really the whole virus research environment for that matter?
Yeah, so I mean, I already mentioned, right? So that originally what is now molnupiravir that got the emergency use authorization against SARS-CoV-2 was targeted against a different virus, influenza, and clinical trial, initial phase 1 trial was planned for 2020. And so we could view it like, okay, the COVID-19 pandemic has really interrupted all of those plans. But I think what it has done is it has set an unprecedented timeline for advance of new therapeutics into the clinic. So the timeline for drugs like molnupiravir or Paxlovid to emergency use authorization is really unprecedentedly short, as is the timeline for SARS-CoV-2 vaccine. So looking back at this, I'm really wondering, would we, without the pandemic, would we now in 2022, early 2023, would we have been in position to run a phase 2 challenge trial against RSV in older adults if we wouldn't have all this knowledge that was gained from use against SARS-CoV-2 about molnupiravir? I actually doubt it. While, of course, resources have enormously been redirected, I think it has really sped up the development of these drugs. And molnupiravir is very attractive because it's so broad spectral. And it also, the pandemic, I think you mentioned the need for rapid testing. And I think I wholeheartedly agree. Now that we see on the horizon efficacious antiviral therapeutics, it makes also sense. We can justify to test. It's not just then we know what the pathogen is, but we can actually then apply efficacious response measures. That, I think, has been paradigm shift. And through the pandemic, what we have done is really we have established an unprecedented infrastructure also of the diagnostic capacity. So the diagnostic capacity we have now nationwide and worldwide is really substantially different from it was pre-pandemic. And this infrastructure now, I think, can be applied and should be applied to make sure that this next generation of antiviral therapeutics is administered to patients early when they do benefit from them the most.
Absolutely. Today we've had Dr. Plemper discussing several things, new therapeutics of note. They are monoclonal antibodies, small molecules, and entry inhibitors. The important thing is that adults will be candidates for some of these agents, and we really appreciate you being with us today. Thank you so much. This is Dr. Forrest Arnold for In Discussion.
Thanks for listening to Medscape's In Discussion Respiratory Syncytial Virus in Adults podcast series with our host, Dr. Forrest Arnold. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out Medscape.com or the Medscape app for show notes, links, and more information on respiratory syncytial virus in adults.